General properties of viruses, part 2. Susceptibility of viruses to physical and chemical agents. Viruses are heat labile, they get destroyed in hot environments, but they are resistant to cold environments. They can survive well at low temperatures. Lyophilization or freeze drying has been used for preservation of various viruses for many years. Viruses live in a pH range of 5 to 9. They are susceptible to radiation. Phenolic disinfectants are poorly virucidal. Oxidizing agents are effective against viruses. Formaldehyde and beta-propiolactone are also virucidal. Lipid solvents can dissolve lipid-containing viruses, that is, enveloped viruses. Antibiotics are not effective against viruses. Viral hemagglutination. What is hemagglutination? Hemagglutination means agglutination of RBCs. Now, some viruses, they have an envelope, and the envelope has hemagglutination spikes. When these viruses having the hemagglutination spikes, they are mixed with RBCs, it will lead to agglutination of the RBCs, which is known as hemagglutination. And when you add antibody against these hemagglutination spikes, then that hemagglutination will be inhibited, and this is called hemagglutination inhibition test. So we can use this property of viral hemagglutination for diagnosis, for identification of various viruses and for their titration. Now, as the RBCs are agglutinated by the hemagglutination spikes, the process of hemagglutination can be reversed by another spike, which is known as neuraminidase spike. Neuraminidase spike is also present on the surface of influenza virus, and it is called receptor destroying enzyme because it will destroy the receptor, which leads to formation of hemagglutination. And this process of reversal of hemagglutination by the neuraminidase spike is known as elution. Viral hemagglutination test में the highest dilution that produces hemagglutination provides the hemagglutination titer, that is known as HA units. The red cells which do not get agglutinated, they settle down at the bottom in the form of a button, while those red cells which get agglutinated, they spread in a shield-like pattern. Use of this viral hemagglutination test is for detection and assay of influenza virus, titration of killed influenza vaccine. Here you can see in this figure, and in certain wells, the RBCs are settled down in the form of a button. These wells are not having hemagglutination property. In 4 wells, the RBCs are spread out in a shield-like pattern, and this is depicting the hemagglutination. Now, viral replication is a very complex process. If you remember, bacteria used to multiply by binary fission or even budding, but viruses do not multiply in such a simple manner. It's a complex process. The process of viral replication has 6 steps: adsorption, penetration, uncoating, biosynthesis, maturation, and release. Now we will have a look at each of the steps one by one. The first step is adsorption. Adsorption means what? The viruses, they are moving about in the environment, and by random collision they will come across cells, various cells of the host tissue. Now, wherever they find a cell which is compatible with them, means which is receptive to them, they will attach on the surface of such cells. And this attachment on the surface of receptive cells is known as adsorption. For example, in the influenza virus, there are hemagglutination spikes. They attach on the surface of respiratory epithelium by binding with the glycoprotein receptors. So the influenza virus has hemagglutination spikes on its surface which bind with glycoprotein receptors of the respiratory epithelium. Another example, HIV-1 virus, it has GP120 receptor on its surface. This receptor of HIV-1 binds with CD4 receptor on the surface of T lymphocytes. So this way, because of corresponding receptors in both the cells, there is binding between the virus and the host cell. This step is known as adsorption. After adsorption, the next step is penetration. In penetration, by endocytosis or varopexis, the virus enter the host cell by forming endosomes in case of non-enveloped viruses, while in case of enveloped viruses, the envelope membrane gets fused with the cellular membrane and then the virus content goes inside the host cell. How does the fusion take place? Entry by fusion with the plasma membrane. Some enveloped viruses fuse directly with the plasma membrane. Thus, the internal components of the virion are immediately delivered to the cytoplasm of the cell. Here an example is shown. The HIV virus has GP120 receptor and GP41 receptor. The T lymphocyte has CD4 receptor and it has CCR5 receptor. First binding of GP120 receptor of HIV takes place with the CD4 receptor of the T cell, and after this binding a conformational change takes place, and because of this conformational change the CCR5 gets recruited in the setup, and the GP41 also forms a membrane insertion into the host cell, and then the fusion of the membranes takes place, and this is how the contents of the HIV virus will enter into the host cell. The next step is uncoating. In the process of uncoating, stripping the virus of its outer layers takes place and the capsid is also removed so that the nucleic acid becomes exposed and it is released into the cell. The host lysosomal enzymes will participate in this process. The genome of DNA viruses is delivered to the nucleus except the pox virus where it is happening in the cytoplasm. The genome of RNA viruses is delivered to the cytoplasm generally. The next step is the step of biosynthesis. In the step of biosynthesis, various portions of the viral components are synthesized in the host cell. Synthesis of viral nucleic acid, synthesis of viral capsid, enzymes and regulatory proteins will take place during this step. And in case of most DNA viruses, the nucleic acid synthesis takes place in the nucleus except in the pox viruses where it takes place in the cytoplasm. And in most RNA viruses, the nucleic acid synthesis takes place in the cytoplasm except orthomyxoviruses, some paramyxoviruses and retroviruses. Steps of biosynthesis. In the biosynthesis, the steps are transcription, translation, replication and synthesis of late or structural proteins. Then comes the step of maturation. In the step of maturation, different viral components that were synthesized in the host cell, they start assembling, and assembly of these viral components will give rise to a virus particle. The assembly takes place in the nucleus in some and in the cytoplasm in some viruses. Example of assembly taking place in the nucleus include herpes virus and adenovirus. An example of assembly taking place in the cytoplasm include picornavirus and pox viruses. Now this is a figure which shows all the 6 steps in one figure. Attachment of the virus on the host cell, that is adsorption, and then penetration, then uncoating, and after uncoating comes the step of biosynthesis which has transcription, translation, replication and synthesis of late proteins. And after biosynthesis, assembly takes place, and after assembly the daughter viruses are released from the host cell. They can be released either by budding or by lysis of the host cell. So this way all the 6 steps of viral multiplication can be depicted in one single figure. The 6th step is release of the daughter virions, which can occur either by lysis, example in poliovirus, or it can occur by exocytosis or by budding. Now what is eclipse phase? Virus, when it enters the host cell, after entry into the host cell till the time viral progeny are produced or viral progeny are released, for this much time the virus will not be demonstrable in the host cell. So from the time of entry of the virus till the release of daughter virions, for this much time the virus will not be demonstrable in the host cell and it will not be appearing in the host cell. So during this time it is known as eclipse phase, and this eclipse phase for bacteriophages is about 15 to 30 minutes, and in case of animal viruses, the eclipse phase is about 15 to 30 hours. And the number of virions released from the host cell is known as the burst size. So virus will not be able to be demonstrated during the eclipse phase. Now what is transfection? Generally a virus or a virus particle, infectious virus particle infects the host cell. But sometimes if only the DNA of the virus or only the nucleic acid of the virus infects the host cell, then also it can give rise to infectious virus progeny. So this kind of an infection in which not the entire virus but only its genetic material infects the host cell, this kind of infection is known as transfection. Now what are abnormal replicative cycles? In certain replicative cycles, when the virus enters the host cell, there are plenty of viruses entering the host cell, and the host cell is not able to cope up with so many viruses. The host cell machinery is limited. It cannot synthesize sufficient number of proteins, sufficient number of components. It cannot give rise to normal viral progeny. But because of this overload, system overload on the host cell, defective viruses will be produced. Some lacking in something, some lacking in something else. ऐसे टेढ़े मेढ़े टूटे फूटे viral progeny will be produced, and this kind of abnormal replicative cycles, this is known as von Magnus phenomenon, in which incomplete viruses will be produced. Abortive infections are another term. In abortive infection, when a virus accidentally enters a non-permissive cell, the cell is non-permissive to the virus, so it will not allow proper replication of the virus. In such cases, defective viral progeny will be produced. Such kind of infections are known as abortive infections. Then there is one term known as latent infection. In latent infection, the viral genome remains persistent inside the host cell. It does not replicate, but it does not cause any harm to the host cell. The host cell survives, and the viral genome remains inside the host cell for a very long period. This kind of infection is known as latent infection. Now, defective viruses are genetically deficient, and they cannot synthesize and multiply on their own. They require a host cell, they require a helper cell and a helper virus. Examples of defective viruses include hepatitis D virus, which requires hepatitis B virus, adeno-associated satellite viruses, which require adenovirus, Rous sarcoma virus, which requires avian leukosis virus as a helper. With this, we finish General Properties of Viruses Part 2.